The Use of Live Animals for Research by Audrey Ruiz, Allison Kidd, Elizabeth Rivera, and Sarah Treichel.
What is/are the ethical questions in the case of the use of live animals for research? Is using any animal in research putting their welfare in danger? Are researchers using animals and following protocol when it comes to their studies? How can it be trusted that these live animals are not coming into any harm?
The main stakeholders and their goals and interests in live animal research. Our first stakeholder is the researchers themselves. They are mainly interested in having effective results and the convenience of using animals when compared to human subjects. As with animals, you have more control and dictation over their day-to-day lives, such as diet and activity level. The animals are our next stakeholder. Their main concerns are survival, a lack of harm and suffering, and their physical and emotional welfare. We also have the community that the live animal research facility is in as a stakeholder. They, as well as the community at large, due to the potential research results and outcomes benefiting a potential number of humans and jobs that the research facility might provide, which leads us into our next stakeholders, which is the employees who may be working with the animals. Their main interests are their physical and emotional welfare when working with the animals. We also have financiers, or the people who are supporting the research projects that are using live animals. Their main goals and interests likely align with the effectiveness of their budget and having usable results.
The ethical assessment of the use of live animals in research. The problem with this is without the proper care and protocols, animals' rights of well-being can be violated when their use is being devoted to research designed to benefit humans. To solve this, the National Committee for Research Ethics in Science and Technology has defined animals as sentient creatures with intrinsic values, and as such, humans' use for them in research reflects our attitudes and influences as moral actors. From this definition of animals, the National Committee for Research Ethics in Science and Technology also defines a set of guidelines in order to ensure the use of animals for research does not violate the animal's well-being. Alternative methods to using animals in research could include using computer and analytical models, the use of which would not impose significant cost to moral values. However, without the use of animals for research, humans would not be able to obtain the same benefits from alternatives. If established guidelines for using animals for research are followed, the animal's rights and well-being are not violated.
There were two peer-reviewed sources that I found, as well as two different perspectives. The first being the perspective of the veterinarians that work with wild mammalians in research and educational purposes. This discussed the protocol that they follow, which includes the ICOOC, which is the Institutional Animal Care and Use Committee, and the ASM, which is the American Society of Mammalogists. Their focus was on the procedure when it comes to housing, humane euthanizing, and much more. On the other hand, there was another survey done in the quality of experimental design, statistical analysis, and reporting of research using animals. In this article, researchers and published papers were examined and asked about not only the research being done, but the scientific method in the case of the purpose of the research, how the research was being done, and what animals are being used. This actually came to the conclusion that the papers and the research published did cause a raise for concern, as not everybody was recording the animals being used, the experiment, and what the experiment entailed, or any much else.
Possible actions and alternatives. The alternatives that we found that we can use instead of live animals was computer models. This alternative using specialized computer models and software programs help to design new medicines by using generated simulations to predict the various possible biological and toxic effects of a chemical or potential drug. Another alternative was cells and tissue cultures, and virtual cell and tissue cultures are easy to follow, less time consuming, and are less expensive. And lastly, microorganisms. An example would be yeast. It has a rapid growth, versatile DNA transformation. Possible solutions that are ethically acceptable. From the 3 choices that we had, we agreed on that the use of modern analytical techniques, data acquisitions, and statistical procedures to analyze the outcomes is any alternative that can provide dependable outcomes. The different approaches would result in minimum involvement of animals in scientific research and procedures.
These are our citations.